Today, I'll be introducing you to 3 new resources from the sea: oyster farming, the harvesting of algae, and the production of medicinal drugs. Since time immemorial, farmers have known how to grow all manner of produce on land. But to farm the ocean, we still rely upon hunting and gathering, and this depletes it. Fortunately, aquaculture, which consists of the controlled reproduction of marine species, is becoming increasingly widespread. Oyster farming has become a major aquaculture activity. Still today, 90% of oyster farming is carried out according to traditional techniques in the oyster's natural environment. Take deep water oysters, for example. In early summer, the male and female oysters release their sexual cells in the water. The sexual cells unite at random and form embryos. The embryos undergo a series of cellular divisions before becoming larvae. Oysters are cultivated by arranging for these free-swimming larval stages to settle on supports called collectors, which are suspended in the sea near the shore. They are harvested between July and September. Collectors consist of shells thread onto rods and, more often, of striated plastic tubes. The oyster brood firmly fastens itself onto it. After about 6 to 10 months, the larvae have grown into young oysters. They are between 1 and 2 centimeters long, and their shells are still very fragile. It is at this stage that they separate from their support. The mollusks are then put into oyster breeding sacs, generally in plastic. This marks the beginning of the fattening period. Depending on the species, this step can last for up to 2 or 3 years. The oysters are sorted periodically. If they do not have enough room to grow, they will become deformed and their shells brittle. Before being marketed, some oyster lots may undergo a final stage known as grow-out. Fattening pond oysters, for example, spend at least 2 months in basins dug out by the tide. These basins harbor an abundant phytoplankton that helps to fatten oysters. In the final fattening stages, oysters turn a greenish color, greatly appreciated by gourmets. The coloring is caused by a substance, merenin, secreted by microscopic seaweed blue navicula. However, shellfish cultivation in a natural environment is a lengthy and haphazard process. Just 1 season lacking all the right ingredients is enough to produce an unsatisfactory harvest, both in terms of quality and quantity. To offset these risks, controlled cultivation techniques have been developed in closed areas called hatcheries. Controlled cultivation in a closed-off environment enables the oyster grower to speed up the oyster's natural biological cycle. In the sea, their reproduction cycle usually occurs once a year, following a thermal variation of roughly 10 degrees. In hatcheries, these natural conditions are reproduced artificially year-round. Oysters intended for reproduction are thrown into a tank of water at a temperature of about 10 degrees warmer or colder. This shock temperature causes the oysters to release their semen. The temperature of the water in the reproduction tanks is rigorously controlled. Further, the growth phase is enhanced with dietary supplements in the form of phytoplankton, microscopic algae, which are also grown in a controlled environment. Another important advantage of hatchery cultivation is that it produces healthy oysters. Oyster hatcheries are spotlessly clean. The premises and equipment are washed with bleach on a regular basis. Any pathogenic infection would immediately result in the scrapping of the contaminated lot. Germs can be brought in on dirty boots, for instance. The seawater that flows in and out of the hatcheries is also a source of microorganisms, some of which can be pathogenic for the oysters. Oysters feed by filtering suspended particulate in the seawater. If bacteria of pathogenic seaweeds are present in the water, the oysters may ingest them and suffer as a result. Filters are therefore installed to trap undesirable microorganisms. During their growth, the larvae are regularly sorted to select the lots that are developing best. These precautions help to guarantee the quality of the shellfish we consume. In a natural environment, however, it is more difficult to control the quality of oysters. Natural breeding grounds are therefore monitored very closely. Whenever a cultivation area becomes too polluted, by fecal matter especially, the adult mollusks are put into special purification plants. They are immersed in clean, disinfected water where they gradually eliminate all the elements that might make them unfit for human consumption. Indeed, the capacity oysters have to retain germs can be reversed. The purification process is very effective. However, it falls short when it comes to viruses. It is very difficult to routinely detect the presence of a virus in a mollusk. Nor is it easy to find out after treatment whether the virus has effectively been eliminated or not. This is a problem of some concern, which the scientific community is striving to solve.
The ocean contains many promising resources, among them algae, or seaweed. Algae are particularly interesting because of their many uses. You probably already know about the virtues of algae bubble baths. But did you know that there are algae extracts in toothpaste and even ice cream?
For centuries, algae, more commonly known as seaweeds, have been harvested and used in many parts of the world. All seaweeds are marine algae. Today, the molecules contained in seaweed are being extracted on an industrial scale. They are used, among others, in the preparation of numerous food and pharmaceutical products. Seaweeds are aquatic plants. Unlike higher plants, they have no true roots, stems, or leaves. Their body is composed of a simple tissue called thallus that floats or anchors itself on the seafloor with its tendrils. Since they have no roots, seaweeds absorb the substances they need simply through their contact with the water around them. These substances are mainly minerals and dissolved carbon dioxide. Light converts carbon dioxide into compounds that are useful for algae, among them sugar. Seaweeds carry on photosynthesis through their colored pigments. The color and diversity of these pigments are among the factors that account for the distribution of the major types of seaweed. Green seaweed, for example, owes its color to the green pigments of chlorophyll. These pigments mainly absorb the red rays of the sun's light spectrum. Red light does not penetrate water any further than 1 or 2 meters. That's why most green seaweeds are found in shallow waters, especially in tidal areas. Red seaweed, however, is not confined to coastal areas. Red seaweeds contain phycoerythine, a red pigment which often masks the green of chlorophyll. This red pigment absorbs the green and blue rays that penetrate the water several dozen meters. Numerous species of red seaweed can therefore be found at great depths. Brown seaweeds, for their part, are usually found at intermediate depths. They contain a brown pigment, fucoxanthin. This pigment absorbs the rays in the median zone of the spectrum, such as yellow. However, the distribution of brown seaweeds is also influenced by the intensity of light, wave movement, and by their resistance to drying. Today, some 25,000 strains of seaweeds have been recorded. Most are of the single-cell, microscopic species that constitute phytoplankton. But some seaweeds can reach giant lengths. Researchers and industrialists are interested in these giant kelp for the hydrocolloids they contain. Hydrocolloids are chemical compounds composed of elementary sugars linked in long chains. What is interesting about them is that they form a gel in water. Depending on their chemical composition and source, hydrocolloids have various applications. Agar-agar, for example, is a hydrocolloid extracted from certain red algae. It is mostly used in the laboratory as a support for the culture of molds and bacteria. Alginates and carrageenans are other families of hydrocolloids. They are used in the food industry as thickening, gelling, and stabilizing agents. They form excellent gels when in the presence of calcium. They are used, for example, to make certain dairy products, such as custards and ice cream. Some types of seaweed can be eaten as is. Their high content in sugars, proteins, microelements, and vitamins make them a choice food. They can be dried and included in various dishes. They are often used in prepared, canned, and quick frozen products. But algae are not only used as food additives, they can have medicinal purposes since they also have the property of concentrating certain chemical elements, such as iodine. Some contain 600,000 times more iodine than seawater. Iodine extracts are used to treat certain disorders of the thyroid gland. The pharmaceutical industry also exploits the many properties of seaweeds and their extracts, alginates in particular. Alginates are extracted from brown seaweed using solvents. After purification, they yield a dehydrated product in the form of a flour-like substance. Sodium alginate powder, for instance, goes into the preparation of effervescent tablets. As it dissolves in water, this salt releases the drug progressively, facilitating dispersal of the active agent in the system. Similarly, surgical dressings used to stem the flow of blood contain calcium alginate, which promotes blood clotting. The applications for seaweed seem boundless. Soon, new natural drugs made from seaweed extracts may well replace today's synthetic ones. Substances with anti-inflammatory, anticoagulant, or antiviral properties are currently under study. We have every reason to be optimistic, for the beneficial properties of many species of algae still remain unknown to scientists.
In labs, a veritable race for drugs from the sea is underway. The goal? Identify all the marine organisms which may contain active substances.
Over half the drugs we use are natural. Many derive from terrestrial plants. However, it seems a good many of these resources have been depleted. We are therefore now looking to the sea for the products to make the drugs we need. Numerous marine organisms contain active substances, some of which can be used in medicine for preventative purposes. This applies, for instance, to certain cold water fish, such as cod. whose flesh supplies oils particularly high in polyunsaturated fatty acids. These acids play an important role in the effective functioning of the nervous system and in the structure of cell membranes. It is believed this fish oil could be used as a dietary supplement to help prevent cardiovascular disease. The Inuit, for instance, a people who live in the far north of Canada and whose diet consists mainly of fish, have far fewer heart problems than other North Americans. It is believed this is due to the action of a polyunsaturated fatty acid in the fish they eat. This fatty acid, known as EPA, combats cholesterol buildup in the arteries. It does this by keeping the cholesterol molecules which are responsible for the formation of atheromatous plaques circulating in the bloodstream. EPA has been shown to decrease platelet aggregation and blood viscosity. This dual action helps to prevent the obstruction of blood vessels, one of the main causes of cardiovascular disease. Fish oils can, like conventional drugs, be put into small capsules. These EPA-enriched capsules can also be used to relieve certain inflammatory ailments. Inflammation is a complex defense mechanism against certain stressor agents. It is believed that fish oil supplements might reduce the symptoms of cirrhosis of the liver and asthma, both inflammatory diseases. There is another type of marine-derived substance that could be used in the manufacture of medication. It is the variety of poison a number of marine organisms produce to ward off predators or to neutralize their prey. Researchers are particularly interested in the toxins released by fish and cnidaria, which include jellyfish and sea anemones. These toxins act on prey by blocking the transmission of nerve impulses in muscle and heart cells. This paralyzes the muscles and sometimes causes an accelerated heartbeat. The researchers believe that small doses of these toxins might be used to stimulate the heart. At even weaker doses, they could act as an anesthetic. The molecules present in marine products are not always actually used. They sometimes serve as models for the manufacture of synthetic molecules. For instance, certain sponges contain substances with antiviral properties. Based on these molecules, researchers are trying to create similar active substances. These synthetic molecules are able to prevent viruses from reproducing in the body. Other avenues are being explored, such as the extraction and synthesis of molecules from sea squirts or sponges to fight the proliferation of cancerous tumors. Materials for prostheses are also extracted from marine organisms. This is the case of that skeleton animal, coral. The structure and composition of coral are so similar to that of human bones that our bodies do not reject it. Coral can therefore be included in the biomaterials used to replace human bones. Implanted in the human body, the coral's honeycomb structure facilitates the invasion of the biomaterial by human bone cells. As part of the ongoing process of tissue regeneration, our body cells destroy the coral as if it were old human tissue. They immediately replace it with new body cells. These implants are well accepted by the human body, since their chemical structure is much like that of mammal bone. Like mammal bones, over 97% of coral is calcium carbonate. Moreover, these biomaterials contain trace elements in concentrations equal to that of human bone. And the trace elements play an essential role in activating vital processes in cells. The oceans and seas constitute amazing reservoirs of life forms from which we can draw medications. Barely 1% of the 500,000 marine species currently surveyed have been studied for their medicinal properties. Unfortunately, many of these species are now threatened by pollution and overexploitation. They may disappear before we have even had a chance to discover their benefits.
For the past few years in oceans around the world, the corals are turning white and dying, the victims evidently of pollution and global warming. Are we now going to destroy what might help cure us?